So, good afternoon, everybody. So now we will start the session on AMR. And it's my great pleasure to introduce you, the two chairmen of this session, Dr. Abdul Salam Ouadraogo, who is professor of medical microbiology at the University Naziboni and head of the Department of Medical Bacteriology and Virology at Saro Sanu Teaching Hospital in Bibo Dioulasso, and Professor Luc Hervé Samison, who is the director of the Centre d'Infectiologie Charles Mérieux in Madagascar since 2011, and the past dean of the medical school at the University of Antananarivo. Can I have my slide, please? So I will start with a short introduction on the roadmap and strategy on AMR. And AMR is one of the top 10 global public health threats and is a major concern, especially in LMIC, with an estimation of around 700 deaths each year linked to resistant pathogen. And ça marche? C'est bon? Vous m'entendez? Yes? And if current trends continue, the results could be with other 10 million deaths by 2050. So AMR is a natural phenomenon. It's a complex issue driven by different factors, especially in LMIC. For example, factors that are very important: the lack of access to clean water, the inappropriate sewage system, the variation in healthcare infection control practices, inadequate provision of antimicrobial and diagnostics, the farming system with suboptimal regulation of antimicrobials, and also the high population densities. So there are different factors that can accelerate the transmission and the spread of AMR. The first one is the antimicrobial overuse or misuse in humans, in animals, and in agriculture practices. The second one is the promulgation of antimicrobial resistant genes in the environment through horizontal gene transfer. And the last one is the transmission from animal to human via direct contact or through the environment, often due to the lack of appropriate sanitary system. So there are also different ways of transmission. The first one is human-to-human transmission through the fecal oral transmission due to failures in sanitation. It can be also due to the healthcare associated infections. And also there are the travel that can be a risk related, a risk that can explain the human spread. And the risk, the travel is a factor of risk for the gut colonization of resistant pathogen. The role of animals and the environment in driving transmission is also very important. There are some transmission from livestock to humans, most likely through the food chain. The use of metals in agriculture can select for resistance. And also it's the importance of sewage and waste processing in environment-human transmission. So you can see with all these factors that the One Health approach is very important to have a good view of the importance of AMR. And it's important to have in mind that One Health the approach of One Health is important if we want to combat AMR. So regarding the urgent needs of action, the World Assembly, WHO have adopted in May 2015 a global action plan to combat AMR, and there are 5 main objectives. The first one is to improve awareness and understanding of AMR through effective communication, education, and training. The second one is to strengthen knowledge through surveillance and research. The reduction of EV incidence of infection through effective sanitation, hygiene, and infection prevention measure also is very important. The optimized use of antimicrobial medicines in humans and animal health, and also make sustainable investment to combat AMR with new diagnostic, new vaccines, new medicines. And according to this global action plan, LMIC have developed their own national action plan to combat AMR. But there are a lot of difficulties for them to implement their plan. And these are due to different factors. First, the weakness of their microbiological capabilities and laboratory systems, the weakness of their epidemiology surveillance network, the weakness of the research, their poor access to innovation, and especially for molecular microbiology. And also there is no funding, especially to implement interventions to combat AMR. So this slide gives you an overview of the AMR programs at Fondation Mérieux. So I will go in some details. So the first activity is to concern the capacity building for diagnostic and lab system. Fondation Mérieux have different network of medical laboratories, and they reinforce the microbiology capacity of this network. So there is a network in Madagascar, RESAMAD, and we have a study with this network concerning the surveillance in human port sector in Madagascar. There is another network, regional network in West Africa and also in Southeast Asia. And in these countries, there are also some grants, country grants from Fleming Fund that can help to strengthen the capacity of these countries in terms of microbiology and surveillance. The second activity is the surveillance capacity building with One Health approach. And you have here the different programs that have been developed so far. For example, in Madagascar, they are well involved in the GLASS program. They report all the resistant pathogens from the human port. There is also in Burkina Faso, in Togo, a project funded by GPIMR, which aims to monitor AMR with an approach of a sampling approach. And there are also different fundings with Fleming Fund, different activities with Fleming Fund to reinforce the capacity for surveillance. For example, there is a Fleming Fund Fellowship Shem in Senegal, or a country grant also in Senegal. All these initiative helps to strengthen the surveillance capacity of these countries. Another also activity that is very important is the infection prevention and control and the antibiotic stewardships. There were in the past a study on the evaluation of POC for better antibiotic prescription. There is an ongoing project concerning the monitoring of antibiotic prescription in Madagascar and in Laos. And also now we try at Merieux Foundation to include the social science component of AMR projects to know more about what are the barriers concerning the perception of antibiotics, the use of antibiotics. It's important to know more about the cultural aspect and to better, after that, implement interventions. And finally, another activity that you know well is knowledge sharing with different courses. Last week, we had the AMR course, the second edition of AMR course. There is also inter-university diploma in Burkina Faso, led by Abdou Salam, which is very important also for the knowledge sharing. So we have also a lot of different relationship with other networks. So concerning the Gabriel network, we try to strengthen the collaborative studies between the South institution, thanks to seed funding. We support also some PhD grant on AMR, and we will try to identify convergence to set up new collaborative studies on AMR. We have also some links with Pasteur Network. They will launch a project which is named SARA Study in different countries in Africa and in Madagascar. And so for Madagascar, we are working now with them to collaborate and to share the same sites of sampling to give a more powerful, to have more powerful results at the end. We are also partner of Avizan Sud. It's a French initiative that develop a network between French institution and institution located in LMIC. And we have developed a research project with different partners in Burkina Faso, Madagascar, Côte d'Ivoire, and in Cambodia, concerning the circulation of multi-drug resistant enterobacteria by using a One Health approach. And we are also partner of the GPIMR network NetEC, which is a network with the objective to enhance the exchanges between all the implementers of the Tri-Circle protocol in the world. Now, concerning the Gabriel network, so we focus on the strengthened knowledge through surveillance and research, and we can summarize the activities by three main pillars, with always the One Health approach. The first one is AMR surveillance, the second one is AMR transmission, and also the capacity building, especially concerning the genomics. So concerning the AMR surveillance, we have worked with different teams. So in Madagascar and now in Paraguay, with the team in Paraguay, to implement the tricycle protocol, which has been developed by the WHO. It's a One Health surveillance with the aims to assess the prevalence of ESBL coli, to get baseline data, and to monitor the different sectors, animal sectors, human sectors, and environmental sectors. And the aim of this tricycle protocol is to assess, in fact, the impact of future AMR intervention before and after intervention, to see what is the impact of this intervention. Concerning AMR transmission, there is a PhD program in Mali that is supported by Gabriel, who the PhD works on the circulation of multi-drug resistance in the different sectors. And there is also a collaborative program between Cameroon, Brazil, and Madagascar concerning the impact of COVID-19 pandemic on the spread of antimicrobial and biocide resistance from wastewater to the environment. And you will have a presentation after my presentation today. And concerning capacity building, the next generation sequencing, the bioinformatics, are now more and more important. And there is a strong need to reinforce this capacity in the members. For AMR, it concerns the global surveillance, and it's important to have early information to know more about the transmission and the spread of AMR, and to know more also about the mechanism of transmission. So just a few words also to give you some information concerning our new partnerships with ANSES, because you probably know that our laboratory, les pathogènes émergents, has been closed in December 2020. And we have established a collaboration with ANSES. It's a French agency for food and environmental and occupational health and safety. And this collaboration will bring the One Health approach, all the approach concerning animals and food. And the main objective is to have a host institution for our staff, for lab activities, and for logistics also, because sometimes we have to order some reagents for our partners, and it's a way to store our reagents and consumables. The other objective is to train students and technicians from LMIC on microbiology, epidemiology, or NGS, and also to targeted appropriate calls on AMR, as we would like to join together to apply for these future calls. And just to illustrate the last training that has been organized in collaboration with ANSES 2 weeks ago. So it was in the ANSES laboratory. We organized training activities on AMR genomics, especially on MinION sequencing, and in bioinformatics in the frame of the Fleming Fong Fellowship SHAME program. So we received 4 fellows from Senegal, and so the scientists of ANSES performed the practical course on MinION, and Emily has organized and performed the course concerning the bioinformatic activities. So the purpose of this session is to strengthen the collaboration on AMR between the members that are currently working on AMR. So that's why after the break, we will have a mapping of AMR activities within the Gabriel members. I have asked to some members that works on AMR to resume, to summarize their activities, to know where are the interests of each members. And after that, we will have a brainstorming session to discuss collaborative opportunities and future work. I think it's my last slide. Thank you.
Thank you very much, Florence, for this presentation, which allows us to have a global view of the AMR activities inside the Gabriel network. We've seen also that she presented all the projects and also the partnerships that exist right now inside this network. If there is a question before we move on to the next presentation. Yes.
Florence?
Yeah.
What's the biggest challenge for us to implement it in a multi-center study inside the Gabriel network? I mean, Brazil, France, Madagascar. My point is it something that we can think in
we will discuss that after during this session. The aim is to discuss how we can have a collaborative project, because for the moment the projects are more isolated. So how we can address the same objective in the different countries.
Yeah,
but it's a challenge. Yes,
I want to put you in trouble. What's the biggest challenge to make it for us when you sit down to and have in mind what's the biggest challenge for make this project work or this study work in a multi-center?
There are many challenges. I think the first one is to find a project that have an impact in terms of public health with all the sectors, because we would like to have the One Health approach. So it depends, as some countries, the heterogeneity of the countries are also present. So we have to take this in consideration concerning the capacity of each countries, but it can be an opportunity to strengthen some countries because we can learn each other. The second challenge is the funding, but we have Laurence that can help us, and she give us before the brainstorming another view of what is possible to in the future to have in terms of funding. So there are not only one challenge, I think more different challenges.
In fact, at the end of this session of this afternoon, there will be a brainstorming, as Florence said before. We will discuss about all of the different aspects that you raised before at the end of this session. So
and the aim is to create a working group we will actively at the end of this session, we need to have some ideas of future project. We don't go outside without any ideas. Don't I know if you don't. And after we will create a working group, and the aim is to meet by Teams, by Zoom regularly to be prepared if we have funding opportunities. Okay.
Okay. Thank you very much again, Florence Brodel. And now we'll move on to the presentation of all the projects that is occurring right now inside the network. And we will have the presentation about a research which is called COV-AMR, multi-center evaluation of the impact of COVID-19 pandemic on the spread of antimicrobial and biocide resistance from wastewater to the environment. And there are 3 countries who participate right now on this project. There is Brazil, there is Cameroon, and Madagascar. So now we have the pleasure to call Mr. Nazareno Scacia from Brazil to present the first part of this project. As you can see, then it deals with the impact of COVID-19 on the spread of antimicrobial. The floor is yours. In fact, Nazareno Scancia received the Bachelor of Science and the Master of Science in Biotechnology from the University of Pavia in Italy. He has been awarded as a doctoral degree in biotechnology and microbiology within the framework of the Mary Slodowska Curie Grant by the Catholic University of Portugal. And he started his scientific career in the field of industrial biotechnology. Currently, he's a postdoctoral researcher at the Institute of Tropical Medicine at the University of Sao Paulo. And very recently, then, he joined the Gabriel Network through a multi-center international research project between Brazil and Africa to investigate on the impact of COVID-19, particularly in the dissemination of antibiotic-resistant bacteria in the environment. The floor is yours.
Thanks for the introduction. Hey, everyone. Good afternoon. First of all, I would like to say thanks for having me here today. It's a pleasure to meet all of you and also to know more about the Gabriel Network as well as the Merier Foundation. My name is Nazareno Scancia, and I'm working as a postdoc researcher at the Institute of Tropical Medicine at the University of Sao Paulo in Brazil. And today, the idea is just to give you a talk about the multi-center evaluation study that wants to study the impact of the COVID-19 pandemic on the spread of the environmental spreading of antibiotic resistance. I'm going to have a short introduction, and some of these concepts were already introduced by Florence. It was a really good introduction. And also I hope we are going to have a really nice discussion, because when we talk about One Health, there are really many things that we should keep in consideration. Now, I'm pretty sure that we all here are aware about antibiotic resistance, and nowadays consider a global threat. And this is due to the increasing of infection disease caused by antibiotic-resistant bacteria. Now, antibiotic resistance occurs when bacteria change in response of antibiotics, making it difficult to treat infection disease. This is just a picture of that I took from this web tool that it was implemented from SCDC to give you just an idea of the increased percentage of antibiotic resistance in Europe, but the same situation is worldwide. As it was mentioned, antibiotic resistance is actually a natural characteristic of many bacteria, but anthropogenic activity have generated environmental reservoir of antibiotic resistance bacteria and antibiotic resistance genes. Now, these red spots that you can see in this image are well documented reservoir of antibiotic resistance bacteria in the environment. And those are the wastewater treatment plants, aquaculture, manure, and also sewage. At the same time, those are also the main path by which antibiotic resistance spread in the environment. Once the bacteria are released in the environment, of course, they can proliferate, and this proliferation can enhance in the presence of specific favorable condition. Now, when this has happened, bacteria can spread antibiotic resistant genes among them. Now, one of the main problems with antibiotic resistance is when we talk about acquired antibiotic resistance. And it sees that many bacteria with acquired antibiotic resistance are of human and animal origin. The ubiquitous character facilitate the dissemination of this of the antibiotic resistance. I really like this picture here because it's just to give you an overview why antibiotic resistance in the environment can be a risk for the human health. Now, nowadays we have to keep in mind that the reuse of treated wastewater has been promoted worldwide, especially for agriculture purposes in those countries under water stress. And in this case, since that this water can have the presence of antibiotic resistance bacteria in this water has been studied, of course, there can be a risk for the human health. here is about the project. So as I was saying before, there is what we call a selective pressure in the environment. So the misuse and abuse use of the antibiotic in the environment can increase the prevalence of this bacteria. So when we, especially now with the pandemic COVID-19, there was registered an increased use of antibiotics. And we should also keep in mind that sometimes the antibiotic is part of the active part of the antibiotic is still excreted in the environment, which can still exert a selective pressure on these bacteria. Now, the project is a multi-center collaboration between these countries, so between Brazil and Africa, and in particularly the study will be conducted by the University of São Paulo, Cameroon, and Madagascar. The objective of the work is to evaluate the impact of the COVID-19 pandemic on the spreading of antimicrobial resistance in the environment, and from the sewage of healthcare facilities receiving COVID-19 patients, as well as wastewater treatment plants. The objective of this work, as written here, are to quantify and detect the presence of SARS-CoV-2 in water bodies. We would like also to compare the abundance of antibiotic resistance bacteria and antibiotic resistance genes, and also comparing the level of antibiotic residues in this water. And then we also aim to analyze the bacterial community, so to have an idea of which bacteria are present there. This is the methodology that we are intent to follow. Please just keep in mind, these sample sites are for the Brazilian team, but the idea is that all the countries will have a same methodology approach. So the idea is to have a wastewater coming from a hospital facility receiving COVID-19 patient. Then we will also collect samples from the wastewater treatment plant and also from a river. In this case, the river will be a kind of control. It's just to compare the anthropogenic, because usually in the river there should be, well, we think there might be a less anthropogenic activities. Once we collect the sample here, and I'm going to, I will show you in the next few slides how we will process the sample. Then we will do some basic physical-chemical characterization of the water, like a measure of the conductivity, the pH, and also to measure the presence of antibiotic and biocide. And then we would like to use this two approach, which is culture dependent and culture independent approach. So the idea is to extract the total community DNA from the water sample, and then screen for the presence of antibiotic resistant genes, and also to perform some metagenomic analysis. Then from the culture approach, we will try to isolate antibiotic resistance bacteria from these different media, and also quantify and see if there is the presence of SARS-CoV-2. And then we will also, like, from these bacteria isolates, we will do phenotypic and genotypic characterization, and also to identify the bacteria. Now, due to some logistic problem, actually, we didn't really start the project in all the three partners. So what we did have a meeting to discuss how to proceed with the work. And actually in Brazil, we have been working in a parallel project, which is not related directly to the Gabriel network. But the idea is to conduct this project that is going to be our pilot study, for which we already draw the protocols that will apply to the Gabriel project. And this pilot study is this one here. Basically, what we are doing, we are trying to investigate the presence of antibiotic resistance in a river that across as slums in Brazil. This is the case of, it's called São Remo, which is a city located in São Paulo state. And basically here you can see that this river is before the slum community. Here, between these two pictures, there is actually a small bridge, and then this water goes inside the settlement. And then here you can see the enter of the river and then just go across the community. Now, when we talk about that, we also keep in mind that actually in this informal settlement, there are not a wastewater collecting system. So basically the wastewater is released directly in the river. And actually in this you can't really see, but in this picture here, you can see these tubes which release the wastewater directly here in the river. Here is how we collect the sample and how you can see how they look like. So basically we are conducting the study once every 2 weeks. We collect this water sample, we keep like a low temperature, we go to the lab, and then we do different things. We follow these two approach, as I was saying before. So we do extraction of the DNA and then we do the cultivable. Now here is the system to do the extraction of the total community DNA. It's basically a pump which is connected to a glass tuber, a glass bottle. So you pour your water and then you filter it, and then you collect this filter, and then from this filter over here you proceed with the extraction of the DNA. And the idea, what we have been doing so far has been like the screening of antibiotic resistant genes by several molecular methods. And we also are being performing okay. And then I thought it was slightly longer than 10 minutes, but okay, I'm going to be fast. And then here is actually from this culture media, we try to isolate resistance bacteria, and then we proceed with the identification. These are some preliminary results that I would like to share with you. You can see that so far we have been doing conventional PCR approach. And what we found now, this is a different water DNA from water struct, and this is our genes for the carbapenems, carbapenemase antibiotics. And as you can see here, there are we detect several antibiotic resistance genes in this water sample. Another interesting thing that we are going to do, we actually start already, but I will not present you this result, and is to analyze the same sample by qPCR, which is a quantitative PCR, to have an estimation of the quantity of the genes, and also with another technique, which is called LAMP. And I'm going to talk about this technique later on. Here, it's just to show you the genotypic characterization of some bacteria isolate. We actually, in total, we obtained 70 isolates. But here I'm showing you just the most representative. This is mean that if you see the red one here are the ones who harbor more than one antibiotic resistant genes. And in this case, you have 3 antibiotic resistant genes. In white one, I'm sorry, but we still need to identify these isolates, and we are doing that. I'm not going, okay, this is just to show you that the idea is to do microbiome analysis, as I already saying. And this is the technology that we are using, which is Ion Torrent. This is a summary report of this technology. It's just to show you that we did run the chip. We have a FASTQ file, and now we are working with these bioinformatic result files to see what's going on there. And yeah, what we are planning for the coming months is to complete this work, and we would like to do this microbiome analysis. The idea is also to do whole genome sequence of the most representative isolate. Well, at least one of the isolate show the presence of 3 different genes, so it would be interesting to see what's going on. And of course, this funding brought us to also the idea to investigate mobile genetic element, because sometimes the spreading of antibiotic resistance genes is due to the presence of this mobile genetic element among bacteria. The idea is also to detect antibiotic resistant genes also by LAMP, as I mentioned before, which is loop-mediated isothermal amplification. And we will talk about this later on, which will be probably an idea to continue to work in this field. And then we will also like to screen antibiotics in this water. And this is a slide with all the partners involved in this project. As you can see here, the people from Brazil, from Cameroon, Madagascar, and also all the institutions involved. This is my last slide. And yeah, I would like to say thanks for your attention. If you have some questions, please feel free to ask. And here I just show you a picture of my lab where I'm working in Brazil, which is LIM 49, and I'm working together with the Professor Silvia Costa. And that's it. Thanks for the attention.
We thank Nazareno Scasia for his presentation. And due to time limiting, we ask one question before move on to the next one.
We can talk.
If there is any question, one question. Yes.
Why did you choose that river in special and not, for example, to use 2 or 3 rivers? Why only one and that special one?
Yeah, it is just due to logistic reasons, but it would be a good idea to have more than one and compare among them. But the thing is that we also choose that one because we think there are not anthropogenic contamination in that kind. Is actually is not really a river, it's a kind of lake. And the idea is to monitor that place because there are not contamination from the environment around.
No, but the river, you showed the one, and the other one
It's-
in a USP is another one, right? That one is
Because
like a lake.
I didn't explain properly. You have a RIA, it's called what it's called a Raya Olympica. So
Raya
basically
Olympica,
you have a river, but next to the river, you have something which is a higher level from the river, where they usually people from the university, they used to have
Ah, okay.
sport.
Okay.
So
Yeah.
this river is kept in a higher level. It's not I don't know the right translation of it's a Raya, it's called in Portuguese. I don't know the English,
Okay.
but it's
Okay. Perhaps you can
Okay.
keep on doing the discussion because it's about the sites in Brazil. As we know, you are from
No.
Brazil.
Yeah,
Is
it's
a
just to
sort
say
of-
that this is higher
Uh-huh.
than the river, so it doesn't come contaminated with the river. So we are sure there are
Okay.
not anthropogenic activity in that site.
Thank you again, Nazareno Scasia, for your presentation. And now we will invite Valérie Donkeng from Cameroon, who will present the same project. And we will talk about what are the activities that have been done there. Valérie is a microbiologist. She began her career in Life and Health Sciences Research Institute School and Health Sciences, University of Minho, Braga, Portugal, and University of Yaoundé in Cameroon, where she obtained her PhD. Over the past 8 years, she works at the National Reference Laboratory for Tuberculosis and Buruli ulcer, Center Pasteur of Cameroon. Now she plays a key role in technical, scientific, and administrative management of the laboratory, and she works now with the Gabriel Network in this study. The floor is yours.
Thank you to have given me the floor. Good afternoon, everybody. Thank you to Gabriel Network to give me the opportunity to present today. I'm going to present on the behalf of my colleague, Dr. Ariane Zwanke, who was supposed to present this study because she's the one conducting the study at the level of Cameroon. So I'm not going to, sorry. Okay, I will not come back to the main objective and the specific objective because it has been presented already with the previous presenters. So I will go directly to the preliminary result that we had in Cameroon, according to this study. So in Cameroon, the study is conducted in Yaoundé, and Yaoundé is the political capital of Cameroon. So Yaoundé has 33% of positive cases of COVID-19. That's why it has been chosen for the study. So the activity carried out so far in Yaoundé or in Cameroon, we firstly elaborate the, we elaborate the standard operating procedure 17 related to sampling, preparation of culture media, laboratory analysis. So those procedure has been shared with the other teams in Brazil and in Madagascar. So we also designed a database called REDCap. REDCap means Research Electronic Data Capture. So it is a secure web application for creating and mapping and managing online survey and database. So REDCap had this advantage that it is secure and fast. It is multi-site access. It had data import function and data comparison function. And also you can export survey results to common data analysis packages. And also for the other partners in Brazil and at Madagascar, their account has been created for this database and share with them so they can use the database. And also another activity was to ordering and purchase reagent and consumable. So we ordered our first reagent on July 2021, and it's only early October that we received the reagent. So in Cameroon, we have two sample sites that are healthcare facility receiving COVID patients. The first sample site selected was Orca site. But when we went there for the implementation, we discovered that this site does not discharge its healthcare effluent into the surrounding river. Because of that, we selected the second site, which is the Jordan Medical Center, for the sampling collection. And we also have a control site, which is a river that receives no pollution link to the hospital effluent and COVID-19. And for this site, we identify 4 sample points with increasing level of domestic anthropic pollution. So for the preliminary data, we firstly prepare a culture media and we evaluate their quality control. So we prepare 4 different culture media as been presented already by Nazareno, and we also assess their quality control concerning the sterility, the specificity, and the selectivity. So for the sampling procedure, we use the ISO 19450 and 58. And samples were collected between 10 to 20 centimeters below water level against the water flow. And the in-situ parameters of physical-chemical, the in-situ measurement, of physical-chemical parameters was measured, like pH, temperature, and chloride level. And after collection, the sample was transferred in the laboratory. So in Cameroon, we perform microbiological analysis, and samples were collected in 500 milliliters of sterile vials containing sodium thiosulfate. And for the SARS-CoV-2 detection, it was also 500 milliliters of sterile water without sodium thiosulfate. So for metagenomic analysis, we collect samples in a vial without thiosulfate of sodium thiosulfate, 1,000 milliliters. And for the quantification of antibiotic residue, we use amber vials of 20 milliliters. So for microbiological analysis, we use the ISO 819090. And serial dilution for the sampling was performed in the laboratory, followed by filtration, plating, incubation, and enumeration of colony. So after that, we perform antimicrobial and biocid susceptibility testing. The SARS-CoV-2 detection and quantification have not yet been performed, so we store the samples at -20 in the laboratory. For the metagenomic analysis also, we filter the sample and then we store them at -18. They will be sent to Brazil for the analysis, as well as the quantification of antimicrobial residue. They have been also stored in the laboratory at -20 to be sent to Brazil for this part of analysis. So as preliminary results, for the measurement of pH and temperature, you can see here that at the Orca Center, the pH value are around the neutrality. So it is between 6 and 7. And for the temperature vary with the sampling according to the sampling period. As far as bacterial load is concerned, Orca Center was the one showing the higher load of methicillin-resistant Staphylococcus and vancomycin-resistant Enterococci, as well as Pseudomonas aureus producing carbapenemase, as you can see in these 2 figures. And also the extended spectrum beta-lactamase and carbapenemase producing Escherichia coli and Klebsiella pseudomonas bacterial load were highest at the downstream end of the control point due to the highest anthropogenic pollution at this point, as you can see here. These are the two control points. So according to the delay that we have to start the activity, we think that by the end of July 2022, we'll be able to have the final report of this study. So the difficulty that we had so far was firstly the delay of the delay between the order and the delivery of the reagent and consumable, which also delayed the start of the field work. And we also had the difficulty with the Orca site. As I showed you before, it was initially selected at the beginning of the study, but it does not discharge its healthcare effluent into the surrounding river. That's why we chose the second site. And I cannot end this presentation without thanks the Gabriel Network who funded this study, and Centre Pasteur du Cameroun, the University of Sao Paulo and Institut de Medicina Tropicale USP, and the Centre d'Infectiologie Charles Mérieux. And for all of you, thank you for your kind attention.
Thank you very much, Valérie Duncan, for your presentation. If there is any question from the floor, from the audience. Yes, Josette?
A small question. So thank you very much for the presentation. What about the COVID-19 results in these locations? So you presented for us the resistance. So you are not also testing the COVID-19?
Not yet.
Not yet.
Not yet. The sample are stored for the study will be performed later. Yeah.
Yes, but I have a question related to this question. What about the timeline? Because in Cameroon, you have different waves. Did you choose a time where there are a high prevalence of COVID-19, or did you take the samples not related to the prevalence of COVID-19 in the country?
Okay, what I wanted to say here is that this sampling that has been done already was for the setup of the protocol, because the three countries are going to start the sampling at the same time. I'm right with Madagascar. Yeah. And
it's a very good question, because you know that the wave are not the waves are not at the same time in each country, so we must discuss. But the good period, in my point of view, is to have it at the highest peak of the wave. We Nazareno?
Yeah, I just want to make a small comment. It is true, it's to set up everything, but the idea is that we start to collect the sample and we will do the sampling for each month. So in this way, then we will compare the data.
Thank you.
Excuse me, can I
yes, from Sofia Vasconez. You have the floor.
It's Silvia Costa from Brazil. Excuse me.
Okay, you can
speak.
You can ask your question, please.
Okay, it's just that we have study regarding COVID-19. And we published recent, and we are going to use the same methodology to perform the Gabriel study. And as Nazareno said, we will have different samples in different months. Then we can compare the rate of COVID-19 in each site, in each country, with the recovery of COVID-19 SARS-CoV-2 from the water. It's just this, my question is just to explain, in fact, what will happen in the project. And the pilot that we made, we already published, that was performed for 1.5 years during the pandemic. So the methodology is ready to start and be performed during the Gabriel project.
Okay, thank you for this information.
Okay, thank you, Valerie. Okay, we will keep ongoing on this multi-center study by listening my co-chair, Luc from Madagascar, who will give us the result about Madagascar in this study. Luc, you have the floor.
Thank you very much, Abdul Salam, for giving me the floor. You know, it is very easy for me to speak because I am the last one. Everything has been already said by the others, and I don't want to be signaled by my friend if I'm too long. So my presentation is about the site of Madagascar. The aim and the specific objective has already been presented by Valerie. Here, I would like to show to you the place where the project will take place. It will be in Antananarivo, the capital of Madagascar, and it is comprised as a low and middle income countries, of course. The situation of COVID in our country is that today, the number of positive cases has been recorded as 43,632 cases. There were 964 deaths. So the case fatality rate was 2.2%. It is important to know that during the last 15 days, there are just 10 new cases that have been recorded. So we are now in a situation where the government has mastered the COVID spread, but the borders have been opened again recently on the 6th of November. So we are afraid now of the introduction of a Delta variant. This is the situation of vaccination. Malagasy people is about 25 million, but there are only, if you can see, 380,000 people who have been vaccinated with one dose, and only around 200,000 who have been vaccinated with full dose. So what were the activities? Of course, these activities have been already presented by Valerie, and it has been done in Cameroon. There is some virtual meetings that we conducted with the 3 countries, and all of the SOPs now are shared in the network. We have to register then for Madagascar with the creation of the accounts, giving the access to users in different countries. And in our country, we have a delay in the ordering of the samples, the ordering of reagents and consumables, because we began to do it in September this year because of the pandemic in Antananarivo, the second wave. And some of those which have been already ordered have arrived in Antananarivo. For example, the PCR enzymes and positive control media from Sigma, consumables, some of the consumables, but there are some which have not yet been delivered, like the PCR primers and probes and DNA extraction kits. So we wait for those different materials to be sent to us before we begin the study. So where are the places that we choose in Antananarivo? In fact, for the healthcare facilities receiving COVID-19 patients, we choose the 2 biggest hospitals in the capital. For those who know, who have been already in Antananarivo, these are the CHU HJRA, Hôpital Joseph Ravang de Naval, and the CHU HJRB, which is the Hospital Joseph Rasset Abefilatanan. In fact, those two centers received all patients infected with COVID-19. So there are different places, of course, the wastewater discharge, the wastewater point before the river, and the channel, Andriantan channel. The channel is directly behind this barrier, and this is the channel in question at the downstream. The second hospital is located perhaps 500 meters from the first one. But the difference between those two hospitals is that for the first, the discharge is sent to an effluent of Andriantan channel, but for the second one, the wastewater is discharged inside the Annecy Lake, which is in the center of the Antananarivo. So we have the water discharge in the hospital, the upstream before it goes into the lake, the Annecy Lake, and the downstream then at the discharge at Annecy Lake. For the control site, we choose again the Igupa River, which is a river crosses, this river crosses Antananarivo from the east to the west. And there are different points that we choose for the sampling of upstream, the downstream number 1, downstream number 2, and downstream number 3, because this is the Antananarivo town. We can see also on this map the situation, the location of the Annecy Lake, which is completely in the center, and the Andriantan channel, which receives the discharge from the Hôpital Joseph Ravagna de Naval. The lake received the discharge from Hôpital Joseph Rasset Abefilatana. So this has already been explained by Valerie. This is the summary of the different activities with microbial analysis, SARS-CoV-2 detection and quantification, metagenomic analysis, and the detection of residue of antimicrobial in the water. So for Cameroon, they say that they began in October, as we will probably begin late December or at the beginning of January. But we have the aim to have completed all the data around July 2022. Our problem then is quite the same as Cameroon, because there was a delay in the start of the field work. They have a 4 months delay, but we had 6 months delay. So at the end of my presentation, I would like to thank the Gabriel Network, Center for Institute of Cameroon, University of São Paulo, and of course my team in the Centre d'Infectiologie Charles Mérieux. And I thank you for your kind attention.
Thank you, Luc, for your presentation. Maybe we have 2 minutes to take another question about this multi-center study, maybe especially in Madagascar, and another to conclude on the network. Who has a question? Because I have a residual question for all the network. Someone has a specific question for Madagascar? Okay, I will go. Maybe,
okay.
No, I have a residual question for all you the three.
Yeah.
I would like to bother you, baby. What was your main hypothesis to conduce the study?
Yeah, it's a very good question. In fact, during the COVID-19 pandemics, there is the use of hand hygiene first, the use of many biocides also. And we would like to know if the data before the pandemic is quite the same as after the pandemic, with the AMR, of course, and also the detection of biocides and antimicrobial in the water.
Okay. Oh, I raise another question. Maybe the last for because we are running after the time. You.
No, yeah. Oh, well, what I understand that the Brazilian study and yours is the same. So when I make the question for Nazareno, Is why, not because it's in Brazil and I know Brazil, but why that river? And you showed another methodology. You have a river that you take samples for different sites. So why you don't standard like the same for the 3 countries?
Yeah, in fact, it's quite the same, because the aim is to have first health care centers which take care of the patients with COVID-19. And the other river is a river site control, which is supposed not to receive contamination from the hospital. That's why we choose the river. As you know, then the river crosses Antananarivo, so all the wastewater from Antananarivo goes inside this river.
Okay, Graciela is begging the the floor.
Because he
Okay.
didn't make the
I'm
question.
not. I don't have the choice. I will give you the floor for 1 minute. Go ahead.
Rapid, very fast. I don't understand really the correlation of how COVID produced the resistance in
Yeah.
the bacteria. So do you have, for example, a data years before of the same place, the same situation, and you can say, okay, 3 years ago, before COVID, we found this. And now with COVID problem, we found this or something
Yeah.
like this. So just the work is wonderful and it tells different things, but I'm not sure if correlation.
Yeah.
Sorry.
Yeah, it's a very good question. And I take the case of Madagascar. All the patients who were symptomatic received automatically macrolides. I don't know if it's the same in the other countries, but that only the fact that all the patients take this antibiotic will surely have a consequence to the microbiota inside the person, and also consequences in the wastewater that are from the hospitals. But from the population, what is the case? That is why we take also sample inside the control site.
Okay,
I don't
very
know
interesting.
if it can. All the patients receive macrolides, and those, of course, who were infected secondly in the hospital with
So if you
pneumonia or
don't
something
find
like that.
COVID in the water, it's not COVID the problem.
No.
That's my question. Okay.
Yeah,
Or
yeah,
is
of
the
course,
time
but
period.
Yeah.
but the let's say the measures that we take around COVID.
Okay, I think we will maybe lengthen the discussion at the break time, but we will take the next presentation, and I let Luc introduce this presenter. Luc.
Thank you, Abdul Salam. Now we move on to the next presentation, and we will invite Milan Milenkov from France. He has to present the prevalence and risk factor and genetic characterization of ESBL E. coli isolated from healthy pregnant women in Madagascar. Milan Milenkov obtained a master's degree in microbiology and immunology in 2008. He's initially worked for Sanofi Pasteur as part of a research team providing research and development support for the production of Bordetella pertussis and Streptococcus pneumonia vaccines. He became involved in the surveillance of ESBL producing Escherichia coli in Madagascar as part of the WHO Tricycle Initiative. He is presently working on his PhD thesis that deals with the identification and characterization of the genetic factors responsible for the resistance to beta-lactams in E. coli isolates from Madagascar. Milan, the floor is yours.
Thank you, Luc, for this introduction. So I will present you my thesis project, which is closely linked to the Tricycle project in Madagascar that Tiaven presented yesterday. So the aim of this is to evaluate the prevalence, the risk factors, and perform a genetic characterization of ESBL E. coli strains isolated from healthy pregnant women in Madagascar. So extended spectrum beta-lactamases, those are bacterial enzymes able to hydrolase a broad range of beta-lactam antibiotics, such as first, second, and third generation cephalosporins and Astronam, and they were described for the first time in 1983. And they have the particularity to be susceptible to beta-lactamase inhibitors such as clavulanic acid and tazobactam. They are thought to be derived from point mutations from narrow spectrum beta-lactamases such as SHV and TEM. And for almost 20 years, those enzymes were mainly carried by bacteria from the genera Klebsiella and Enterobacter, and they're responsible for hospital-acquired infections. And then since the beginning of the 21st century, we observe a dissemination of those enzymes in the community, and this time they are carried by E. coli, and they are not more related to the SHV and TEM families, but they are mainly from the CTXM extended beta-lactamase family. Global human carriage of ESBL producing enterobacteria is estimated to be around 16%, with significant regional variations, as you can see on the graph, with around 10% in Europe, for instance, and more than 70% in Southeast Asia. Those bacteria are responsible for more than 50 million infections per year. And they are not only resistant to beta-lactams. Very often they carry co-resistances to other classes of antibiotics. And this makes the treatment options quite limited. And the first choice for treatment of this kind of infections are carbapenems, which in its turn leads to the emergence of carbapenem-resistant strains. So the reservoir of ESBL producing enterobacteria, as we already saw it from other presenters, are humans, warm blood animals, livestock, and wild animals, and the environment. And in this context of the World Health Organization proposed a project, a One Health project called Tricycle. And the main objective is to measure ESBL E. coli prevalence in the main city of a participating country with 100 samples from ESBL E. coli positive blood cultures, and then 100 healthy carriers, and pregnant women have been chosen for the study. The food chain is presented by 240 chickens per year, and the environment is represented by 6 sampling campaigns performed on surface water upstream and downstream of the capital, human wastewater, and slaughterhouse wastewater. So in Tricycle, Fondation Merrill tried to perform an extended Tricycle project because we included 7 laboratories and not a single one. 6 of them are clinical bacteriology laboratories, being part from the RESOMAD network, and they dealt with the human component of the study. 3 of them are situated in the capital city, Antananarivo, and 3 in Provence from the cities of Mahajanga, Ampatuan-Razak, and Tomazin. And then the CICM Research Center in Antananarivo was in charge of sampling and analysis of the food chain and environmental samples. So the main objective of the thesis is to evaluate prevalence of ESBL E. coli and then to perform a phenotypic and molecular characterization of the isolates, antibiotic susceptibility testing, field determination of the phylogeny groups, sequence types, Fimbria serotypes, and so on. And then to identify the resistance and virulence genes carried by these bacteria, point mutations, the plasmid content, and try to find if there exist genetic links between isolates from different origins. And finally, we will perform an analysis of clonal dissemination of isolates from the 3 sectors only in the capital city. So this is to present quickly the characteristic of the study. So here I will present only results from the human component and only asymptomatic carriage, because we were unable to obtain a sufficient number of ESBL E. coli positive blood cultures. So we had 492 women, 166 of them were positive for ESBL E. coli, and we obtained 305 isolates from those. And then we performed antibiotic susceptibility testing, which was used to establish an antibiogram susceptibility profile for each strain and we only sequenced strains that presented distinct antibiotic susceptibility patterns. So we sequenced in total 168 isolates from pregnant women. And then we analyzed the risk factors associated with the carriage, and we observed big differences between the different cities, with only 12% of the pregnant women being positive from the city of Tomazin, and more than 65% for the city of Ambatondranzac. And the only risk factor for carriage we identified was the wet season. And the wet season in Madagascar is also the warmer season. And our hypothesis here is that it may be explained by the fact that conjugation and plasmid transfer between bacteria are facilitated at higher temperatures and humidity. So if our hypothesis is true, it means that global warming trends could even worsen the situation in the future. And we had 34% of carriage for all those pregnant women. And other risk factors as age, level of education, access to drinking water, toilets, electricity, or recent intake of antibiotics were not associated with a higher carriage. Then, yeah, sorry for this slide, I forgot to include some points in the animation. So all our isolates were resistant to cefotaxime, and we detected a CTXM enzyme in all of them. Then we had an isolate resistant to carbapenems, about 25% resistant to gentamicin, more than 80% resistant to fluoroquinolones, and 70% to sulfamethoxazole, with a very good correlation between phenotypes and detected genes. And then the isolate which was resistant to carbapenems carried New Delhi methyl beta lactamase 5, and this is the antibiotic profile of this isolate. In red are on the left, the three circles in red are carbapenems, and the one on the right is mesalinam. And at these diameters, the strain is completely resistant to those antibiotics, and it was only susceptible to fosfomycin, colistin, and chloramphenicol. So these kind of strains are really of a big concern when present in invasive infections. So about phylogenetic groups, most of our isolates belong to commensalism-associated phylogenetic groups A, B1, and C, and in a much lesser extent to D, B2, and F, which are associated with extraintestinal infections, which is consistent with the commensal nature of our isolates, because all these pregnant women were asymptomatic. Then we constructed a minimum spanning tree based on the sequence types. And as you can see, isolates belonging to a single sequence type were found in all the cities, or most of them. So it means that we are not in presence of a clonal dissemination of VSBL E. coli carrying strains, but it is more about independent strains belonging to a big diversity of sequence types. We performed also geographical mapping of isolates from the capital, because for the capital we had the addresses of the pregnant women just to the quarter level in order to preserve anonymity. And once more, we found no geographical clustering of the sequence types in the capital. So it means that ESBL E. coli does not carry about rich and poor people, and it is evenly disseminated in the capital city. Then we constructed a phylogenetic tree based on the single nucleotide polymorphism, and we observed a main group of phylogroups A, and as you can see in blue, and as you can see, we had 7 isolates in blue which clustered more with isolates from D1 and C phylogenetic group, and D, sorry, phylogenetic groups. And it means that we are probably in presence of phylogenetic group A isolates which begin to diverge genetically from fuel group A. And the other interesting thing here is that we found a lot of strains in which CTXM5, CTXM location, the ESBL enzyme, was predicted to be chromosomic. And it is really worrying because bacteria can lose their plasmids in the absence of selective pressure, which is not the case when the enzyme is integrated on the chromosome. Finally, we tried to analyze only strains who shared less than 10 SNPs of difference in different cities in order to see if we had some clones disseminating. And it was the case, but quite limited, as we had some strains which shared less than 10 SNPs difference and present in different cities, probably due to intercity travels. Then we analyzed the virulence genes content, and we separated we grouped those virulence genes in some categories: adhesins, iron acquisition associated genes, protectins and invasins, toxins, and other virulence genes. And we observed quite a low number of virulence genes per strain, which is consistent once again with the commensal nature of the isolates. And as you can see on the right, isolates from phylogenetic groups A, B1, and C, commensal phylogenetic groups carried much less virulence genes than the invasive phylogenetic groups B, 2, D, and F. Then we analyzed the plasmid content of the isolates, and most of them on the right carried a single plasmid replicon. But some of those strains carried up to 4 different plasmids. And the most prevalent one was INCF, which is consistent with global epidemiology, which was what was more interesting is that we detected a high proportion of strains carrying INC-Y plasmid, which is really unusual. INC-Y is described as a really rare plasmid in E. coli, and we found more than 20% of our isolates carrying INC-Y. So we decided to perform nanopore, long-read nanopore sequencing in order to try to reconstruct the whole sequence of those INC-Y plasmids and see what they really carry. And we were able to do it. As you can see, the 38 strains carrying INC-Y plasmids shared a significant degree of sequence identity with some deletions for some of them. And the reference strain carried the tetracycline resistance gene TET-A. It had 2 copies of it. Then the efflux pomp QNRS1 conferring resistance to fluoroquinolones, the ESBL CTXM15, the penicillinase TEM1B, then genes phosphotransphorases conferring resistance to aminozides, and sulfamide and trimethoprim resistance genes also. And interestingly, when we blasted our sequence against the NCBI database, it returned more than 99.9 sequence identity with an INC-Y plasmid, which has been described in Finland and is of avian origin. And this particular bird species does not go to the southern hemisphere. It is limited to the northern one. So it is very unlikely that the same INC-Y plasmid was found in Madagascar in this bird species. But it means that wild birds can be involved in ESBL dissemination also. So in conclusion, we observed a very high prevalence in healthy pregnant women in Madagascar. 35% places Madagascar among the countries with the highest prevalence in the world. The wet season was the only risk factor associated with carriage, and our hypotheses about global warming trends worsening ESBL dissemination have to be explored more. And we observed a great genetic diversity of the isolates coming from the different sampling locations. Most of them were from phylogenetic group A associated with commensalism. And in consequence, we found virulence genes among those isolates. So the most worrying observation is the detection of a carbapenem resistant strain carrying NDM5, and particularly in the absence of a strong selective pressure, because carbapenems are used in Madagascar much less than in developed countries because of their price. So it is worrying to find these carbapenem resistant strains. And in perspective, we will now analyze the results from the whole genome sequence of the isolates from animal and environmental origin, and then compare isolates from the three sectors in order to try to identify genetic links among them. And I would like to thank the CICM Madagascar team, because they did a great job about collecting the samples, analyzing them phenotypically, and then sending them to France, as well as people from the ReSaMAD work who did the same for the human component. And then the INSERM IAM team from Paris, my thesis directors, Laurent Serment-Lefebvre and Antoine Andromont, and all the staff there supporting me. And of course, people from the Fondation Mérieux, especially Juliette Bryant, Florence, and Hubert Tans, Laurent Raskin, who showed me Madagascar, and Emilio Westel, who was involved in most of the bioinformatic analysis. So thank you for your attention.
Thank you very much, Milan, for your presentation. Now it's time for the questions. Any questions? Yes, Florence?
Yes, thank you, Milan, for your presentation. I have one question concerning your, I think not last slide, but when you mentioned that the plasmid was mainly found in a bird in Thailand, does it mean that the birds carried also the ESBL E. coli? Do you have some?
Yes.
Yes.
The bird carries ESBL E. coli, and this E. coli strain carries the same plasmid, which on its turn carries the same ESBL enzymes.
So that means it's another way for transmission?
We think so. That wild birds are involved in dissemination of ESBLs globally.
Okay.
Yes.
Okay. Thank you for your very wonderful work on TreeCycle. I noticed that due to the diversity of clones, you think that it's not, that's my opinion too. It's not an epidemic of one clone. Why not conclude that we can have an epidemic of plasmid due to the pressure of antibiotic use? Because we have seen that you have mainly one plasmid which is very frequent.
You're talking about INC-F or INC-Y?
Maybe INC-Y, which carried the most gene for ESBL.
We are not sure about it. I mean, INC-F was the most prevalent plasmid.
Yeah.
It's the case worldwide.
Yeah.
So it's not INC-Y. But what's your point?
Yes, I did. I don't have the result, but it's one hypothesis for me, because sometimes your work is wonderful because you deeply investigate about the diversity and the distribution of clones in different areas and from different people. But we have mainly this finding in many countries. If we are exploring ESBL distribution, we don't have the same clone. That concludes us that it's not an epidemic of clone, such it can be for others infection. We could have one microbe which is spreading out from hospital or outside. But here we have diversity, very high diversity of many of clones. it could lead to conclude that we have a diversity of bacteria, and due to the pressure, bacteria are willing to get plasmid.
Okay, so you mean that it is more a dissemination of a single plasmid?
Yeah, maybe
Yeah,
an epidemic of
it is a
plasmid.
hypothesis that has been already described before, because some plasmids are adapted to a given species or certain strains from this species. And it's the case of INCF plasmids particularly. And then a lot of those plasmids carry toxin, antitoxin systems. So if the bacteria loses the plasmid, it will die. And this prevents the bacteria from losing the plasmid. So these are those successful plasmids that disseminate globally. And it is probably the case with our plasmids also.
Okay.
Okay, we would like to thank again the presenters of this session. And now it's exactly 3:30. We are sharp on time. So it's time for the coffee break. And we will be back at 4. Okay, thank you very much.
Hello. Hello, hello. Okay. Everybody is okay to resume. You could resume this passionate session on the topic of AMR, and we will continue the discussion about identifying convergences and cooperations on antimicrobial resistance with the Gabriel network. And for that, we'll call Jean-Yves Madec. Jean-Yves Madec is doctor in veterinary medicine who holds a PhD in public health, postgraduate on MR in human medicine, molecular microbiologist and research director. His research interest focused on molecular genomic and epidemiology of MR in One Health perspective, and he is an active participant, a leader in European transnational research projects. He is the coordinator of the ZAPAT network monitoring MR in clinical animals in France and provides senior level guidance, expertise, and support to the implementation of One Health NAP on MR in France. Jean-Yves Madec, floor is yours.
Yeah, thank you very much. So hi everyone. Again, I'm very happy. Thanks to Fondation Mérieux for inviting me. I'm very happy to be with you face to face as well. I had the chance to participate to the Lebanese meeting of Gabriel. It was nice. So thank you again. So this presentation is not really a presentation. I think it's probably most likely the start of the discussion. Florence asked me to present some of the work we have conducted with different countries. Just to start as a start, I mean, to provide some examples of some common work we could have done, to provide some inspiration, some questions we have addressed with different countries. And then afterwards, after the session or after the presentation, the mapping of your activities on the AMR issue, the objective is really to work together. So this is not a presentation, this is just the start of the discussion. But of course, it could be an idea to present what we have done, and this is the objective. So many things have been already said on AMR, so I will not go too much into detail. So just to summarize, but you know that who we are. So we're, as said by Florence, we are a national agency for different issues, food, environmental issues, any risk of humans in their daily life. That is what we're working on. We are based in Lyon, and we also have laboratories. That is quite uncommon for an agency, because you just need meeting rooms and experts usually. But we have, this is a story from history. We have laboratories conducting research and surveillance activities. And is it this one? Okay. Yeah. And in the field of EMR, we, on the surveillance issue, we contribute to the surveillance at different levels. The first one is, of course, at national level, and we coordinate the surveillance of the monitoring program called RESAPAT on EMR in animals. This is our main focus. But from animals, of course, we expand to the environment, to the connection with the human sector. But our main task is in the non-human sector. And we also coordinate, this is the recent work we have done in the frame of a EU JAMRAI action. It's a joint action from the European Union to coordinate the surveillance of EMR in animals at the European level. And recently we were also appointed at FAO reference center on EMR. And this is also a way to contribute to surveillance of EMR at a more global level. In terms of research, we have different topics, but the main topic is on molecular genetics and phylogenomics. So what we do is to work on One Health issues, transmission, how the genetics is behind all of this, and how the different sectors are connected, interconnected, I mean, animals, humans, and the environment. This is not the only topic, of course. We also do some more basic research as well, but this is our main task. And this is what we do for the French authorities as well. They want to know where EMR is, what are the trends of EMR, especially in animals and in the environment, but also what are the possible connections. Because as long as we're talking about One Health, we need to know what are the main routes, because the second objective is to put some intervention somewhere. And because money is limited, because time is limited, because the resources are limited, it's highly important to target where we have to intervene. And this is also something we can bring into the discussion in order to screen everywhere, which is probably not that useful, but to see, and we don't know much in a One Health perspective, where to act. And this is what we want to discuss also with you. Just a couple of results on surveillance of EMR at national level. This is the trends we have in France on animals, on EMR in animals. So you can see that you can achieve very nice objectives and results. This is the trends of resistance to fluoroquinolones in E. coli in animals. Of course, this is in France. In France, it's in Europe, and there are many challenges that we don't have compared to other countries, of course, but it is possible to decrease the resistance to antibiotics in the different sectors. And what we will try to do is to have the same outcomes at European level. So this is the network we have built during the last 3 years called ESVET, which is reminiscent of ESNet in human medicine. And the objective is to collect data from all Europe or the European Union in animals and to decipher where are the trends, in what direction, where are the main challenges in terms of AMR in animals. In terms of research, okay, this is a key word for us, One Health. We conduct research in the One Health perspective. So again, starting from animals, but we are very much connected with the human sector. And all these sectors are connected all together. The human side, the human sector is connected with animals, with the environment. We all live in the same world. But we probably have to take, to be cautious on the fact that everything's becoming to be One Health now. Everyone is talking about One Health, and One Health is everywhere. And sometimes there is kind of lost. We lose a bit the meaning of One Health. It's not because One Health is everywhere that everything is connected with everything with us in the same manner. So it becomes highly difficult now to really came out from this One Health objective and really to retarget the main pathways where AMR is transmitted from subtopics, subsectors within the big 3 sectors. Okay, so just a few examples. This slide is quite, this work was good. Thanks, Milen. We're not aware together that we were presenting the same work, but this is very, very nice work. It basically shows that the SPLs are a very large challenge, large threat, and more importantly, that we are not, ASPLs are not equally distributed over the world. And this study was a 10-year study between 2000 and 2010, or a bit more. But now, if we were, for instance, here in Bangkok, instead of Annecy, all of us, or almost all of us, would be ESPL carrier. This is quite amazing. In France, the prevalence is not that high, it's but 7 to 8%. But in certain part of the globe, the carriage, the healthy carriage of ESPLs is quite amazing. Of course, you can find ESPL, and especially ESPL E. coli in domesticated animals. There is no species, no need to look for another species that do not carry SPLs. You will not find a new thing because ASPL has been described everywhere. And of course, in many other sectors, globally the environment. Yes, in wild birds, you can find ESPLs. You can find also carbaphinamases in wild birds. It's not that easy to connect wild birds to the real world, I mean the terrestrial world. But there are some studies. I was discussing with Milen. There was some. We can look on the study, but I remember a study in Australia where they deciphered the pathway or possible likely route of transmission of VIM4, I think, between cats, wild birds, and humans in the same city. And so to say that also for the next discussion, it is important to really consider that any kind of data is important and metadata is important. Just sequencing everything everywhere is not a good option. It's part of the response, but remember also that if you find the same thing in two different places, it does not mean at all that it's connected. Could be, but could not be. And you need, to solve this issue, also you need metadata. You need, so we for the next working group we will have, it's highly important in my view to try to find relevant settings from where we could expand interpretations, not sequencing everywhere in all sectors and get a lot of data without a good metadata. So it's highly difficult to connect ESPLs in birds and ESPLs in humans, for instance. But in certain circumstances, you can do that. But you can see here that ESPLs are really widespread. So a couple of studies. This is different studies we have conducted with different PhD students or scientists in Tunisia. The objective was to find or to describe ESPLs in animals. The question was, do we have ESPLs in animals? Where? What are the prevalences? Are ESPLs more prevalent in cattle, in pigs, in poultry, in whatever? What are the genes, the clones? Are they similar to what we observe in human medicine, the human sector? So this is kind of question we can have. But this was also said by the previous presenters. AMR can also transfer through food. So this transfer through food is also a major One Health issue. You can address this type of questions. This is something we have done also with Tunisian colleagues in the Bicêtre Lagoon, where there are 4 hospitals charging effluents in this lagoon. And not that surprisingly, we have found a lot of multi-resistant bacteria and Enterobacteriaceae producing carbaphinamases in seafood, I mean, in oysters, in mussels. So probably this highlights that those filter organisms were contaminated by multi-drug resistant bacteria directly coming from effluents. So this is also a kind of local study, but from this study you can draw hypotheses, and then if you want to intervene, you can say, okay, there's something to do with the effluents from the hospitals, there's something to do with the food chain, and you can build upon that. So this was an interesting study. Another one was conducted by another PhD student, Lydia. She went to me and told me, okay, I would like to look for AMR in the food chain, and I have the opportunity to look in kebabs. It was in Algeria. She said, okay, that's good. I like kebabs. So please go on. So she bought 200 kebabs, and then she asked me, what do you think ESBL are within the kebab? Is it the salad, tomatoes, onion, bread? I don't know. I just eat kebab. So she did a little mix of all of this. So we don't know from where ESBL is coming from. So you can imagine that the second study could be to separate, to think. But well, what we have discovered is that 25% of kebabs were contaminated at the surface with ESBL. So it means that you directly eat ESBLs, because she stopped the transmission chain because she had the freezer here and then the gloves and so on. So so this is first information. And the second information, this was in the city of Béjaïa in Algeria. Another information is that when we go to Genomex, we looked at the clones and the plasmids, and the surprise was that we found exactly the same clones depending on the district within the city. This pink circle indicates that we have found the same clones here, and those clones were different in another part of the city. So you can think about hypotheses. Could be the same vendor, the same knife, the same, could be the water, could be the salad of this vendor and not the other one. So this is a more global study. It's the first study, it's a pilot study, but we have never done that because the PhD was over and it's what's okay. But we could have had follow-up of this study, looking at different pieces of the kebab, different district and so on, and look for common sources or different sources. It was a nice study. So we have seen that the plasmids play a major role in the spread of ESBLs. It's a review we have done recently, and as Milan was saying, INCEF plasmids are really widespread. Certain ESBL plasmids are considered epidemic. It means that you find them everywhere or very frequently. Some others, like INCY, are not, but sometimes you can find a pop-up of INCY1 plasmid that you may want to understand. And this is a study with regard to plasmid spread we have conducted in Brazil in companion animals. You can see here that those plasmids, INCY1 ST12, INCY1 ST12, INCY1 ST12, the same plasmid in different bacteria and different dogs and cats in the street. So this is to answer the question of the plasmid spread. Sometimes you have clonal spread, sometimes you have plasmid spread, sometimes you have both, combination of both. But in this case, it was in Sao Paulo. There was no connection between, apparently no connection between the dogs and cats. There were different bacteria, species, not species, strains, and the same plasmid. So this is interesting. Why? Another plasmid story was this one. This is a plasmid we have identified just after the cholesterol crisis, when the new gene of the MCR family was discovered in China. Everyone was looking for this gene everywhere. So we have done the same in France, and we identified this gene MCR1 on this plasmid, INCHI2ST4. Okay, that's purely descriptive. In France, MCR1 is located in this kind of plasmid. But because we had colleagues from Tunisia who have seen that, we asked them, okay, maybe you have MCR1 as well. On which plasmid? This was a question. So look at this plasmid. Oh yeah, the same one. So then why? Because there are so many plasmids spreading ESPL or MCR1. Because in China, it was not this plasmid, it was INC-I2 plasmid. And in other parts of the world, there are also different plasmids carrying MCR1 genes. And the reason is because you can see here, all the birds were originating from France. So this is most likely, this is the best hypothesis we have. Probably the Tunisian colleagues have important birds and the plasmid. So this is also a question you can address. The impact of the food animal trade on the spread of AMR. This is another type of question. But if you want to address this type of question, you have to design something to go in that direction, to say, okay, we look for imports. For exports, we take 2 countries, 3, 4 from the group, and we see whether there are some connections, and then we can interpret, because we know the metadata, we know the trades, the imports, importation, exportation, and so on. This is another kind of study we can conduct. And this is a nice story with our Lebanese colleagues, Mohammed and Hiba, who are 2 PhD students, a doctor now. And the objective was to report the situation of ESPLs and other genes, carbapenemases as well, in different sectors. So we started with animals, the environment, then the human sector. And yes, we did more or less the same as in Madagascar. We described that Lebanon was full of ESPL genes everywhere in cattle, but also we have found OXA48 in milk directly from cattle. This is unusual. And also ESPLs and carbapenemases, carbapenemase producing enterobacteria in water sources in the environment. And then we went to more advanced techniques, introducing whole genome sequencing, of course. And from this study in the poultry sector in Lebanon, we also found that there was one sampling point at one slaughterhouse. Because it was easy, it was the way the design was done. And we have found many, many different clones. So as soon as you know that, because when you are thinking about slaughterhouses, it could be hospitals, could be the same. You can think of nosocomal or clonal spread within this setting. It was not the case. So the problem was not at slaughterhouse. The problem is in the farms, because sometimes you have a clonal spread in slaughterhouse, so you need to intervene in the slaughterhouse. But this was not the case. Well, more or less the slaughterhouse should be okay, but the strains were very much distributed in all the farms. And another point of the part of the study we have done is this interesting work in food workers. We have sampled food workers in pastries at T0 and T6 month, people working in pastries, the same ones, 80 people, around 80 people. And the question was, do they carry ESPLs? Do they carry carbapenemases producing bacteria? And how is the carriage? How long is the carriage? Is the same, we will, the question was whether we will find the same clone or the same plasmid at T0, T6, or whether they will lose the clone. How is the dynamic between intake of the clone and loss of the clone? Is it more a clonal spread within the pastry, or more plasmid spread, or a combination of both? And from this study, what we have probably the feeling is that clonal spread is much more efficient in that kind of setting than the plasmid spread. And also the second point is that when we have found, for instance, many clones of the ST131 sequence type carrying the same ESPL gene and many other common features. But when you go to the whole genome sequencing, it changes dramatically. What we thought was identical 5 years ago is not identical at all. Because if you have an E. coli differencing by 40 SNPs from the other one, even though it is the same sequence type, same ESPLs, it's a different story. So it's highly important to go into this kind of details to to try to decipher the real pathways of transmission. Okay, so this is my last slide. So just to talk, just to start, just kind of studies, just to report to you what you had the opportunities and the great pleasure to do with colleagues. I think we share convergent objectives. And the keywords could be epidemiology, One Health, transmission, and impact at global level, not only in France or in Europe, but at global level. We already have common partners, and our Lebanese colleague is an example. We shared probably interest in training, capacity building, we've seen that, co-supervision of PhD students. So the question is how, this is written there, and I thank you, Florence, for the kind introduction in the booklet, because this is exactly your sentence. How to enhance local applied research to improve laboratory-based characterization of AMR, and I added, and to solve relevant scientific questions. Because it's highly important to identify which question or which issue we would like to address. And not say, I want to sequence everything in my country. This is not the objective. So thank you for the time and for your attention. So from One Health, from heart to real life, it means that in all the best papers, you will find this graph with arrows between the different sectors everywhere. Everything is connected with everywhere, but most probably some connections are more important than others. So please find them. We have to find them because we don't have too much resources. Money is limited, time is limited, and AMR is growing. So we don't know much. So this is highly important to identify the main important questions. And those questions may be different from one country to the other one, because the setting is different, your situation is different, the way where you are located on the globe is different. So this is also what is really interesting and exciting in working all together to find a common problem with different ways to solve it in different countries. And I hope we'll have a very interesting discussion. Yes, and yeah, soon, and you will not go out of this half of here without any smart and ideas yet. So thank you very much.
Thank you. Thank you, Jean-Yves. I think that we still have 5 minutes to have maybe 2 hot comments about your wonderful talk and fitted to our objective to identify convergence and cooperative ideas. Someone has specific comments about, because the thing here is to comment and to identify ideas and maybe project on which we could together work. Someone has comments. We have, we can have 2 comments, and the floor is to,
So that bird, it starts in France, and but do you know if it goes to every continent?
Well, actually, those birds you are talking about, this plasmid between France and Tunisia. Those birds are not flying. They are important at, I would say, 1 day old. Chicks, yeah, and raised in Tunisia, but the source is in France. So this is not a question of flying; it's a question of just important these chicks. To Tunisia, that was the case. No. We can sell to the whole world. Yeah. Because for me it was strange because Brazil also exports. Yeah, exactly. Yeah. Yeah. But I would not exclude that also birds can fly from France to Tunisia for any other reason. That's not this.
True. And this part of One Health too. One Health, one world.
the mother. Oh, my birds can fly to just to get the
Okay.
chick back.
Okay, Florence, go ahead.
Is there any investigation in France to see how these birds?
Yeah, we have done that
Okay.
last year. We have published no, not 2 years ago. We have published the paper last year. We decided to answer the question: is are birds really a reservoir of ESBLs? So how could you address that? So we decided to collect feces from birds at one sampling point because it's easy, and we went to a center for where you collect birds suffering injuries, where people bring the birds. So we discussed with the colleagues and so on. Okay, when the bird is entering your center, you collect the bird immediately or not, or in the couple of hours after entrance. But you may know that those kind of centers are crowded with a crowded activity, especially during spring and summer. And well, we did the work and the molecular work and so on, and we have found prevalence of 25% of the birds carrying SPL. So it's not possible. It's not possible. What happens? And we went back to the records, and actually they were too crowded. There was too much work. They could not sample the birds at entrance. So they were waiting for a couple of days. And during those days, the clones were circulating. There were an internal spread, and the number of clones were limited. One main plasmid was circulating everywhere, so we could not have the answer. but we had the answer. We could provide recommendations to those people. That's okay. What you do afterwards is to release the birds in the nature. So you are just an amplification center for ESBLs. So it was important to highlight the procedures, the hygiene measures they could set up in the center. But we didn't have the answer on the prevalence of ESBLs in birds, but most probably the prevalence is low. I would not expect that much because it's more incidental capture of ESBL genes from feeding habits or something, but it's not that much. But it's interesting.
Okay, many thanks
for
your presentation. Okay, we'll go on the next presentation, and it will be done by 9 presentators, if I see through, mapping on the AMR activities within Gabriel Network. And we will have some who will present physically, and others will be in Zoom. And for beginning, I would like to call Sarah Eyango from Cameroon. We will agree to give 5 minutes for each presentation. Since we have 45 minutes for 9 presentations, we'll have 5 minutes, and we will discuss at the end of the presentation. So a little bit presentation of Sarah, who is currently the scientific director of Institut Pasteur of Cameroon. She's a big flag of mycobacteria studies in Africa. It's what I could say very few about her. Sarah, if you are ready, floor is yours.
Okay, thank you very much. So as Valerie, I'm presenting also on behalf of Dr. Ariane Zwanke, my young colleague who is on maternity leave. So I will just quickly follow the canvas sent by Florence. So talking about AMR activity in Cameroon, so what Ariane put is that AMR is 100% of current research study. I can say it's okay, a part of some years studies, but conducted not directly in bacteriology lab, but in the neglected disease platform. that saying, what I can say is that when we are comparing other activities in in the institution, virology lab, mycobacteriology lab, or malaria, I think the IMR or the bacteriology activity is the less developed one. So we have to really stress on these activities. Even if she put that it is 100 activities, yeah, is the less developed activities. So the type of activities are the surveillance, but I can say it's more routine collection of data, more less than not really surveillance. So that is the targeted pathogen, mainly the enterobacterial order. But also we published last year a good paper on Neisseria gonorrhoeae, the resistance showing the increase of ciprofloxacin resistance within the 10 years of data collected in the lab. So yeah, the One Health component, including wastewater like variation, but also animal, but more in chicken is also included. So method is still to be developed, more classical susceptibility testing and molecular characterization of some marker, and we are still going to implement sequencing. Next. Okay, thank you. So concerning quickly the priority research, I think Valerie has presented the one we are conducting with within the Gabriel network with Brazil and Madagascar. And we have just received a support from Pfizer Corporation. This one will come to complete the one we are conducting with Madagascar in order to help us to not send sample outside for genomic analysis, but to integrate in our CITES activities. And we will also add the modeling approach to help to have and tools not directly linked with human, but which will help to estimate antimicrobial consumption in healthcare facilities. This fund is coming to support or to complement activities going with the previous one funded by Fondation Mérieux. Main difficulties, I think it is same difficulties that Florence already raised, limited funding, but I can also add limited human resources. We really need to strengthen laboratory staff. We need to integrate multidisciplinary approach to see how we can set an IMR network in our country, because we are not really part of GLAS. We are not beginning with the tricycle activities. So these have to be also implemented. But as you mentioned, we are also part as a Institut Pasteur International Network, part of SARA program. So it will also come to reinforce the organization of the IMR network. I think it's almost 5. Thank you very much.
Thank you. 4:30 is very on time. I will call Firdausi Khadri from Bangladesh about the same topic. I think that I don't need to introduce Professor because he's very renowned.
Yeah.
Professor, you have the floor.
Yeah, thank you. Actually, we prepared a few slides from Aideshi to talk about our interests. We are mainly on the host side, not on the animal end, but learning from what's happening today. we should be in the bigger picture of One Health. So the things that we're doing right now is service and research, and which takes up 40% of our current activities, and mostly with human health. And so we are doing a lot of disease surveillance, respiratory and enteric pathogens, both in our own communities in Bangladesh, but also among the Rohingyas, together with Valentina. And we also support other facilities that do not have the access to testing for AMR. You may understand, as you know, we have people from many countries in the world. It's not that every lab has the facility. And our etiologies of interest until now is strep pneumoniae, Klebsiella pneumoniae, Acinetobacter, Staph aureus, and enteric bacterial and other viral pathogens, Salmonella, enterotoxigenic E. coli, Vibrio cholerae. We've been working with Florence on E-Tech and E. coli. We have a project, it's in publication now. But we're also interested in fungal etiology because we know it's a major source nowadays, Candida especially, hospital-acquired infection. So we are always on the host side, mostly. So we would like to actually work more on the One Health component. We do a lot of patient study, healthy contacts. Whatever we get in patients, we try to look at healthy contacts, and we find many things, same things in a healthy person. And also the environmental spread, especially looking at sewage water in urban areas and in rural areas. But urban population is one major reason for spread of infections and also AMR. So our capacity right now is conventional microbiological methods and RT-PCR tested for pathogens. We're doing a lot of RT-PCR testing, not only for COVID, but other pathogens. And we are moving very quickly to automated YT2 testing for AMR, and also MICs, which we are already doing. And we are looking forward to more capacity in genome sequencing using bioinformatic tools, using Ariba. This is one request I put to Foundation Merieux and the Gabriel Network to increase our capacity. And our research priorities on AMR include the LARI project that you heard about today, low acute respiratory infections among the Rohingya population. And we also are working on the correlation between COVID-19 infection and enteric pathogens in another collaborative study with Nottingham University. So when there are COVID patients, we're also looking at the nasopharyngeal swabs and also stool to see what the microbiome is actually doing. And also Klebsiella pneumoniae. Jannat over here has a new project on Klebsiella pneumoniae, and both invasive and non-invasive, using hospitalized patients, but also healthy controls. We have, we usually enroll healthy controls from the same area that we have patients. So for the project in Cox's Bazar, our healthy controls always come from the same area, same site. And for Klebsiella, it will be in the urban slums because this will be a Dhaka city-based study. And we have a global health fellow, another one who's working on respiratory, bacteria, and fungus among adult community-acquired pneumonia patients and healthy controls. This is being set up. Next slide, please. Oh, sorry, I forgot I was not. So our main difficulties encountered are that, we did not have automated systems, but I always take a big step forward. Small steps come later. We are investing in both the Bactilert and a Vitek2 system and bioinformatic tools, and which I will be dependent on Gabriel linkage. And we are also establishing an advanced laboratory information management system within our facilities to have a very good analysis of what is coming from where and sources. And we want to expand our expertise and also share our expertise, because IDESHI stands for Institute for Developing Science and Health Initiatives, which means we always train other people all over the country. And as ICDDRB in my main institution does, IDESHI follows the same role, and we do a lot of capacity building for other institutions. We do not have access to any of this or do not have the infrastructure. And always reagents and funding is a problem in developing countries, everyone understands. And so I think we're very grateful to the Gabriel network for allowing us to participate in this. I've got how many people? 4 people here, and also one from ICDDRB. So, and it's very good to sit in one platform and listen to everyone talking about the same things and the same problems and solutions. And I was very intrigued by the talk just before me, and from Jean. We always pronounce wrongly, and to see that, the facility that he has in his institute can be used by all of us for our analysis, and not only to go to that institute, but also set it up in our own institutes, because I think the plasmid analysis, the genomics is very, very important. And so I think we're still not going into chickens, but we wouldn't mind, because that's how everything comes in, the ESBS and the MBS and everything. So I'm very grateful to to Valentina and my microbiologist who's just joined us, Shanchita, who has many plans, and Jannat is always there, and also Farhana from ICDDRB. Thank you. I don't have, but I'm very interested in what's happening.
Okay, we'll move on. Next presenter from Bangladesh, who will join by Zoom, Shakil Ahmed. If he is connected, we'll give him the floor. Shakil, welcome.
Yes, hello. Good night from
Shakil.
here. How are you?
Fine, you? Hear from
Okay.
us?
Yes, you can see my screen.
We are reading your emails.
Okay, sure. Now you have seen this.
Not the presentation.
Sorry, I'm
Yeah,
just starting.
okay. It's fine. It's fine. Okay.
So actually, I was supposed to be in this conference in the Gabriel meet, but unfortunately, because of my restrictions and COVID problems and government restrictions, I could not join in time. That's why I am presenting from my
Please,
home.
Shakil, we didn't hear better from you. Could
now you are hearing?
Yes, it's better. Thank you.
Thank you. So I am from Bangladesh Institute of Tropical and Infectious Diseases, BITID, a collaborative laboratory with Foundation Meru, and the lab is called Rodolfo Meru Laboratory. And I was supposed to be there in this Gabriel meet, but because of the government regulations and visa restrictions on movement of the government officials, I could not join. That's why I'm presenting my paper from here. We in the BITID is doing the AMR surveillance that started in 16 and is fully going from 2017, and it occupies only 15% of our total activities, because it's a large research plus educational institute now, and it has got a multifaceted activities. And BT is actually a part of the collaboration with the National Surveillance System, is one of the 10 central sites of National AMR Surveillance Program continuing since 2017. And IDCR is the central authority, and they are monitoring the AMR surveillance centrally, and all of the data that are generated are entered in the GLASS network. We are mainly working with stool and urine. There are 5 pathogens, 5 specimens like CSF, sputum, blood, stool and urine, but because we do not have that much of indoor facilities and surgical or gynecological ward, we are mainly focusing on stool and urine. And priority pathogens are especially Enterobacteriaceae C, Escherichia coli, Klebsiella, Enterococcus, as well as ESBL and Pseudomonas. And priority pathogens in the stool are Salmonella, Shigella, and Vibrio. And because this is a delta region and we are in the meeting point of the river and sea, it is a very good area for growth of Vibrio. So Vibrio is very prevalent in this area. And we work with this urinary tract infection and diarrhea. These are the case definitions. Sample mainstream urine samples were collected in sterile container. This is I'm not going to discuss. And we do the antibiotic sensitivity testing by Stokes distribution method, and we use the control strains for standardization. And all the sensitivity tests done by distribution method, and we follow the CLSI guideline. And these are our results from various years, 2017 to 2021 till date, stool and urine. And these are the main important pathogens, Salmonella, Shigella, Vibrio. You can see the percentages of them. And also in the urine, E. coli, Klebsiella, Enterococcus, Pseudomonas, some Staphylococcus and Staphylococcus apophyticus. These are the organisms that we are getting here. And we are doing many research also regarding AMR surveillance, study of entomicrobial resistance of bacterial pathogens isolated from patients in diarrhea that has been published. And we are also published by the urinary tract infection, but we do not have any One Health component now, but we're very much interested to start with the animals as well as the environmental. And we are also working in close contact with the SIVASU, that is the Animal and Veterinary University. And we are doing some collaborative activities to start the One Health project also. But we have many difficulties because it is a specialized infectious disease hospital. So we don't have all the wards that is responsible for these infections. Mostly the surgery and gynecological ward is not there. So we are not getting that much of wound swab and pus. We do not have ICU facilities now, and that's why blood culture or nasopharyngeal swab or a meninges CSF is not available that much. In the outdoor, we do not have STD cases. That's why a very important point of interest is missed. We have the Bactelart and Vitek facilities, but because we do not have that much of blood culture, bottles is not available. Vitek cards is also not available. That's why we cannot use them properly. But now we are trying to collaborate with some other institutions like MSF in MSF hospitals working for the Rohingya population in Cox's Bazar. We are working in collaboration with them, and we are supporting them with the blood culture through the Bactelart and Vitek systems. And especially in the government hospitals, we sometimes run short of reagents, and especially the anti-seras for the specific identifications. And one thing that is lacking here is the dedicated research workers or students. But we are supporting the MPhil microbiology students, those who are doing the post-graduation in microbiology in the Chittagong Medical College under Chittagong Medical University, and we support them in their research activities. And they are working in Mycobacterium, AMR, and other different activities. Thank you very much. That's all from my side.
Thank you, Shakil. We hope we will keep connected for the brainstorming in 30 minutes. So we'll move to another presenter and be back for the question. So I will call Professor Kuriba from Mali if he is connected.
Hello.
Hello, Professor.
Yes, good afternoon. Can I have my presentation, or should I load my presentation? I don't know how to proceed.
Maybe you have to share your screen. Oh, okay.
Is it? Yes, good.
It's fine here.
Okay, thank you very much. I'm Burama Kuriba from Centre d'Infectiologie Charles Mérieux, Mali. It's my pleasure to share with you some of our antimicrobial resistance activities. We have around 30% of our research activities are antimicrobial resistance. And we do diagnostic and surveillance studies here. And the targeted pathogens are bacteria, mainly Enterobacteriaceae, and also the TB, MDR, and Metis, Staphylococcus aureus. And also we have antimicrobial resistance surveillance on the One Health component, mainly on animals and environment. And I will share with you some of our objective for this part of our activity. The methods we are using are the classical methods, and for the bacteriology, and we are actually using also the VTEC and PCR. We are actually trying to set up the PCR to monitor the antimicrobial resistance. The next slide, can you put the next slide? Yes, we are actually doing a study on at one of the main hospitals. It's Gabriel Touré, which is the second main hospital in Mali, and we are going to do a study on the in intensive care units and operation room. And the objective is to assess the bacteriological incidence of nosocomial infection and antimicrobial resistance in patients hospitalized in this intensive care unit of the hospital. We're expecting to determine the incidence of bacterial infection associated with the care in this intensive care units, and to determine also the bacteria responsible for the nosocomial infection, to test the sensitivity to the antibiotics of the isolated bacteria, and also to monitor the molecular marker of antibiotic resistance in this setting. That will have probably to improve the quality of the care in this intensive care, because we are suspecting a lot of infection in this intensive care, and that probably will reduce the morbidity and the mortality to this intensive care. Also, we are going to do another study, or we are actually doing a study. Next slide is on the occurrence, source, and prevention of antimicrobial resistance in Mali, following the flow of AMR genes between human, animal, and environment. This study is ongoing and in collaboration with University of Helsinki, with Kaisa Huka, and we are doing this study also with Burkina Faso, Benin, and Helsinki. The objective is to determine the gene responsible for antibiotic resistance and the mechanism of dissemination between the environment, animal, and human. The expected results are identifying the genes responsible of antibiotics resistance and the mechanism of the dissemination among animals, human, and environment. And that will have increased our knowledge on this AMR diffusion in Mali, and to improve the control strategy of antimicrobial resistance. That is the our two main activities. We have a lot of antimicrobial activities. Because of the timeline, I'm only presenting these two activities. Thank you for your attention.
Thank you, Professor. Keep connected for the discussion at the end of the session. And we'll go on by inviting Moser Ams from Lebanon for his talk. Thank you.
Good afternoon. I would like to thank Madame Florence Pradel and all members of organized committee. Fiche verte. Pardon. C'est compliqué, hein? Bon. Alors, AMR is 70% of our current research activities. Type of activity, diagnostic, mechanism of resistance, and molecular typing. Targeted pathogen, Mycobacterium tuberculosis, carbapenem-resistant Enterobacteriaceae, plasmid-mediated colistin resistance, non-fermenting gram-negative bacilli, Pseudomonas, Burkholderia cepacia, and Stenotrophomonas maltophilia. Our research activity on AMR has a One Health component. We are working on AMR in the human, animal, and environmental sectors. Method technology use, standard antibiotic resistant testing, molecular techniques. Priority research project, assessment of the contribution of the environmental pollution by antibiotics, residues, and resistance bacteria, and the spread of bacterial resistance aimed at developing strategies to minimize contamination of the environment. Main difficulties encountered and way to overcome. World Bank says the current Lebanese crisis is one of the sharpest depressions of modern times. The currency has fallen by more than 90%, demolition, purchasing power, banking system is paralyzed. You can see other model of a problem, not technical problem now. Fuel shortage has crippled normal life, affecting essential services, including hospital, transportation. The country is experiencing a multi-sectoral breakdown in the delivery of basic services like water and power. Medicines, including antibiotics, are in short supply. Laboratories are running out of kits needed for blood tests. Patients are delaying going to health facilities because they cannot afford care. Healthcare providers, including doctors and nurses, are leaving the country in search of better salaries abroad. Thank you for your attention.
Thank you very much for keeping time for this, for better discussion at the end. We will hear from Madagascar by Luc.
Yeah, thank you very much, Abdul Salam, for giving me the floor. So for Madagascar, the Centre d'Infectiologie Charles Mérieux is conducting researches, and amongst those, AMR represents 16%, because we conduct also a lot of researches on leprosy, on mycosis, on schistosomia, and tuberculosis. The type of activity on AMR is surveillance, and the targeted pathogens depends on the type of project. For the first one, the tricycle one, the targeted pathogen is ESBL Escherichia coli. And for the Triumph, which is the recent project that we just began, we have ESBL Escherichia coli plus Carbapenemas producing Enterobacteriaceae. Does your research activity has one health component? Yes, because on those two projects, we deal with the human sector, the animal sector, and the environment. And the methods that you use is hemoculture first for tricycle 1, because it has been already presented before. We collect all the data of positive blood culture, especially then about ESBL E. coli, and sooner with CPE. And we perform also a lot of cultures plus antibiotic accessibility testing and the whole genome sequencing with the invoice of samples in France. But sooner we will have a sequencer, and we will be able to do that. The main project that we are conducting right now is the TRIUMPH project. The aim of this project is to improve the tricycle protocol. And the specific objective is to upscale to national monitoring, the detection of ESBL E. coli, but also the CPE. And we would like to put in place the WGS pipelines for One Health surveillance. For tricycle, there were 7 laboratories which work with us, and for the TRIUMPH, we will upscale that with other regions, and we would like to have 10 laboratories in each different parts of Madagascar. So the expected results that we would like to have, of course, is the generation of data on CPE prevalence in humans, animals, and environment, but also the WGS analysis of the isolates from this different reservoir. And the TRIUMPH extends the tricycle protocol to a second region in the other countries. But as I told you, for us, the human part will we will have 3 more laboratories with 3 other regions. For the environment and for the animal, we will have 2 more sites of samplings. Then the last expected result that we would like to have is the trainer approach with the materials, protocols for further extension of the project in the future. So what are the difficulties that we encountered? In fact, there was a delay in the release of protocols for the detection of CPE, talking about this TRIUMPH project because of the pandemic, COVID-19 pandemic in Europe. The laboratories with which we work didn't have finished on time the release of the protocols, because 3 different teams work on it. One team work on the writing of the protocol of the detection of CPE in animal, one team in human, and one team in the environment. But we are now at the phase of validation, and we have the hope to begin the samplings from January 2022. And that is the presentation of Madagascar, and I thank you for your attention.
Thank you, Luc. Next presenter will be Rosana Gillen Freitas from Paraguay.
Good afternoon. Thank you for Gabriel Network for the opportunity to be here. I'm Rosa Guillén, and I work at the microbiology department of the Instituto de Investigaciones en Ciencias de la Salud from National University of Asunción, Paraguay. Our main activities include 70% of research in antibiotic resistance. We do surveillance in collaboration with a local network that has been put in place a decade ago. with all the reference hospitals of the Asunción and the department, the Central Department of Paraguay. We work with targeted pathogens like methicillin-resistant Staphylococcus aureus, mainly causing infections in children, but also work in Enterobacteria resistance to beta-lactamine and carbapenem, mainly in E. coli. We focus in variability, genetic variability, and virulence profile of these pathogens. In E. coli, we work with Shiga toxin producing E. coli. We have worked in several projects that concerning cattle, and also we have worked in water, searching E. coli in rivers that cross Asunción, that is the capital city of Paraguay. And also we work constantly in the human component. We use conventional culture and PCR and DNA sequencing-based methods, but the laboratories that collaborate with us, they have Bitech and Bactalert and also Malditov technologies. Our priority research is to implement the global tricycle surveillance systems for E. coli in the protocol. So we are now presenting the protocol to the ethical committee of our institution. We already get some of the reagents, and we aim to begin the protocol in 2022. The main objective is study this pathogen like indicator of organisms of the phenomenon of expansion in antimicrobial resistance in human, animals, and environment, as you have heard before. Maybe it's this? No? Okay. I don't know what I did. Thank you. Okay, thank you. Thank you very much. What are the difficulties we encounter? Basically, the lack of funding is common for all of us, but we are fighting that, and we are currently get some grants, local grants that will allow us the first reagents to start the project. And we also have prepared proposals to get researchers granted for our university in the next year. The other difficulties maybe is they get to the permissions to access to the city sewage sources. That is not easy many times. And we think that we can have access to outlets, sewage outlets outside the system, because it's common in our city that they have this kind of outlets in public places near the river that cross our city. And thanks, that's all for me.
Thank you very much. We will go to Brazil. We have two presenters. Firstly, Nazareno, sorry, Nazareno.
Hello again. I'm presenting for my supervisor, which is Professor Silvia Costa. And the main activity in our lab, EMR in our laboratory is the 80% of our research activity. And we mainly do surveillance, diagnostic, and also treatment, including stewardship. As a target pathogen, we work mainly with the ESBL and also resistance to colistin and fosfomycin, and also with healthcare associated infection, such as carbapenem resistance, VRI, and also MRSA. We do have a One Health component. We mainly work with human and environmental samples, and now the idea is also to start with samples from animals. And we mainly work, we do metagenomics. We also have a conventional molecular technique. And we also do whole genome sequence, also for the One Health component. At the moment, we would like to focus on this project, which is the aim to implement the LAMP technique, which is a PCR-based technique. And for this urinary tract infection in the primary care units, as well as in public hospital. And we will also try to test this technique in the environmental samples. This is the idea of the project. So the object, as I was talking before, the idea is to implement this technique, which is really, it could be quite cheap and really easy to use. And it can also implement it with a colorimetric method in order to investigate the presence of antibiotic resistance gene on-site analysis. We are planning, the idea here is to implement it, to develop a protocol, particularly for antibiotic resistance variant, which are mainly spread in Brazil. This is the BLA-OXA 148 and also the SPM variants. But the idea is that if we manage to implement this, to develop this colorimetric tool, the idea is also to develop this technique for other genes. And what I would like to underline here is that the same rationale could also be used for other infection disease, such as tuberculosis and also other kind of genes. And as a main difficulty, of course, we have, it's about the budget funding for scholarship, and also sometimes with to receive a consumable. If we need something for outside Brazil, for instance, it will take a really long time. And that's it. Thanks.
Thank you very much. We will listen the last presenter by Zoom. If she's connected, if she is connected, Anna, she's connected. Okay, welcome, Anna, over here.
Okay, sorry. Okay.
Okay, the floor is yours.
Okay, thank you very much. You can see my screen? Yeah.
Yes, but
Yes.
it's not in Japurama.
Not in full screen.
Yeah.
I think it's arriving.
Yes.
Yeah. Thank you.
Okay. So thank you very much to be here again today. Unfortunately, I thought that is only 2 slides, so we have prepared only two slides. And regarding to our RMR, currently at the lab, since the beginning of the pandemic, we still working with RMR, but it's only represent 8%, more or less 10% of our activity, as we are now working a lot with COVID. So we work in this presentation. You can we do present only the result from the surveillance project that we have developed together with the Unifesp, is a university at state of São Paulo. That the person that lead this project is Professor Ana Gares. He's an infectologist, and we work together for 15 years, more or less. So in this project that was supported by the Bill Gates Foundation, we performed the surveillance from the 5 different regions in Brazil. And we work in different pathogens such as Enterobacteria, Pseudomonas, Acinetobacter, Enterococcus, Salmonella, and wherever many pathogenic organisms. And we work also in this project, we collect samples from different regions from Brazil, from health individuals, and also from food producing animals, such as poultry, cattle, and swine. And for develop these projects at the lab, I'm working bioinformatics. So I developed many tools for analyzing the data. I'm a geneticist. So we use our platform for our sequencing platform, and we perform a metagenomics analysis from which replicates from this sample from this four different types, and we perform more or less 300 metagenome samples. And using this data, we are able to identify many microbial resistance, antibiotic microbial resistance. We are able to perform the assembling for different genomes from the genus until the species. It was 2,900 more or less different organisms, and we are able to identify many different resistance. This result of these two works are submitted for publication at this moment. And what the main of this project is to identify the spreading of RMR in Brazil in different regions. Our main problem for other than money support, financial support for the project is always access and collections for the samples. And this is regarding the metagenomics, but we are also working for many years from assembling genome sequencing and assembling many different genomes from different organisms. I thought it's only to talk about metagenomics, but I collect some papers that we have published in the last 2 years regarding from the different clinic. Most of them are related to a clinical and molecular description from different bacteria, mainly Klebsiella, that's very common in Brazil, but also with birds. I saw the presentation before here in Brazil, and Enterobacter, and trying to identify a new resistance and a new genomes here circulating in Brazil. So it's more or less, thank you again, Gabriel, and thank you for Florence. And we can collaborating for trying to discuss a little bit in the next section.
Thank you very much, Anna. I would like to propose to have the last presenter which will give us an overview of funding opportunities before going to the brainstorming discussion, because this presentation will be very helpful for the next session. So if the connected colleagues are still over here, they could keep connected. Thank you. Laurence from France.
That's right.
Welcome over here. And the floor is yours.
Great, thanks.
Yeah, for 5 minutes for an important topic. Thank you.
An important introduction.
Yeah, thank you.
Thanks so much. So first, I want to say that I'm quite impressed about all the work presented during this first part of the meeting. And so this is really the great place to introduce, as Laurence mentioned, so way of collaboration, project, portfolio of projects, not on a one-shot approach, but a long-term approach. So the idea here is to present you in a two-step. So just to keep the spirit as before. So a long-term a long-term approach, which will enter in gap analysis that we will see after. But first to present some opportunities. So the only, I was quite frustrated when we discussed with Florence, because the opportunity are not perfectly aligned, and we are in a transition period at the European level with the African Union. So there is still a lot of things coming in before the end of the year, so which led the opportunity for project to be there. So the first, I will try. Yeah, it's not a good light. So the first is a topic presented by the EU for Health. So it's more on a European focus. And unfortunately, only 2 among the Gabriel Network countries could be eligible. So because there are 3rd countries which could be eligible, but not already mentioned their interest in participating. So where AMR is one of the key topics around training activity, implementation, and also best practices. I will go on the 3rd one. So Horizon Europe presented a topic regarding COVID and so on. So with pandemic preparedness, so which could be aligned to some of your activities that you already presented, reacting to the COVID, but in a more general approach. So how we deal with pandemic, taking an X virus or bacteria. So leading to best practices and how to deal with. The 3rd one, and I think it's really an important one, as some of you already contribute in some EDCTP. So the new one is EDCTP 3, but more global health. You will hear more global health terminology. And AMR is one of the important topics that will be included in the future topic. So hopefully we could have before the end of the year, before Christmas. So and for sure we will inform on the current topic. So the second one, and really more in the long-term approach. So this is really to come from your needs, I would say as a gap analysis. So really from where you start and where you want to go in terms of impacts, not only on saying research capacity or whatever, but really concrete. As Jean-Yves mentioned, it's important to have a focused approach, either as methodology that could be raised among partners, but with impacts which could be country specific or area specific. So which I mentioned more as a general approach, and to have really the topic that you are interested in with we can say the right consortium, the right funding, and the right place. So this is also how I would like to support the team in this way. So there are also, I would say, 3 other structures progressing. So the European Partnership for One Health related to AMR, and I'm sure that you will be involved in the future of this. So focus on AMR. So, but the structure will be established in 2022. So meaning that the topic will come in 2023. And for ERA for Health, so it's another structure dedicated on health, which raise different countries. So it's a really national approach, and as a joint forces. So with the AMR topic for sure included in it. So IHI, it's another, but more related to industries. So in the same, unfortunately, in the same area. So the IHI will be launched before the end of the year. So hope with some topic beginning of next year. So this is the end, and after you will receive some documents to present objective topics and where it is feasible to build on. Thanks so much.
I didn't see anything about Asia over here. This is all CTP, will be all European for the Africa part.
Yeah, the core is Africa for sure, but I saw in the regulation that some additional countries could be added. But it's really on a one-to-one approach. So meaning that if you have a pilot, there is the idea to replicate in a partnering country. So it is feasible in the regulation, it is feasible.
Thank you, Laurence, for
Thanks.
this talk. I think that you are beside us for the next session, which will be the last but not the least. And we will open the discussion, the brainstorming
Yes.
session.
I think it would be nice if all the speakers can
In
be
front of-
close here. We can sit on the
Yeah.
of the scenes,
Okay.
and all people that are interested by the discussion, please come to form the working group, and also all the participants online, Hannah, Dr. Shakil, Kuriba, and other people. If you want to join, please open your camera as well in order to see you, and do not hesitate to intervene.
Thank you, Laurence. But it seems that we are sleepy in this conference room. Maybe we will stand up. We'll stand up to be sure that we are not sleeping. We can stand up, everybody. And try. Maybe we'll try to touch a lamp. To be awake. Wake up, wake up. Thank you, thank you.
Some exercise.
Yeah. Okay, everybody is wake up. Okay. I think that we will begin the discussion, and I would like to quickly introduce the discussion, because
before, who will take some notes? Who would like to take the notes of the report?
I think we will appoint 2 reporters to have the main ideas to share at the end of the session. Okay, I let my co-chair choose for the.
Perhaps we will ask the youngest man or woman in this room to be the reporter. Yeah. Thank you, Graciela.
Graciela, come on.
And who would like to be with her? Who would like to be with Graciela?
Zanat, you can.
Okay, thank you.
Thank you so much. We can. Yeah.
Okay, so yeah, you can take down all the discussions so that at the end we will have anything to report to Florence.
The main ideas.
The main ideas.
Not
not all the details, but the main ideas.
Yes. A second one.
Is there anyone else?
Kevin.
The youngest.
Do you want?
Okay.
Okay.
Would you? Perhaps it's better you place yourself there. You move. Yes.
Okay. Thank you, everybody. I think that the presentation you can come in front of. Don't hesitate.
Come on. Yes.
Come
don't
on,
be.
come on.
Don't be shy.
Naive. Yeah. Welcome. I would like to quickly summarize the session. This session around AMR underlined the arm of AMR, especially in low and middle income countries, which many causes scale up the burden. And we must have new approaches which be aligned with the main pillar of national action plans. Fortunately, Fondation Melier has many initiatives as such as capacity building, many programs and networks. And within these networks, we discuss deeply about their activities. I think that many of the teams have show here their capacity in terms of human research, capacity in project development. And right now, I think that we have to discuss how to build something which could gather us around this topic, to work together on antimicrobial resistance, to strengthen the network, to draw the other teams from behind to front of, to help each other to build a solid teams to implement AMR project in their team. And I think that I can summarize our session. I won't enter in the detail of the presentation of each country. It's not needed here. The very important to see to discuss is in which field we could have a project which could gather the maximum of teams of the network. In regarding of the reality, in regarding of the One Health approach, and in regarding the experience of what is already done in each team. Thank you. If my co-chair would like to add something.
It's okay.
Yeah.
Perhaps to begin, it's interesting to have the specialties of all the laboratories. How many of us deal with the human part of the AMR? Can we raise our hand? Yeah, so there, yeah, two-thirds, 4/5 of the laboratories can perform surveillance or researches on AMR in human. Those who can deal with animal, those who can deal with animal, yeah, there are not so much laboratories with animal.
Luc, you
yes.
have some people
Yeah,
who
Ana
are
Teresa.
raising their
Okay,
hand.
1, 2, 3,
Yeah.
4, 5. In the environment. Also, okay. So we can see from that situation that all of us could more or less deal with all the 3 sectors. Okay. So that is the first situation that we have. So what is the kind of researches that we can conduct together to fight against AMR? Is it a surveillance? Is it for example, to implement tricycle in every country, or to search for another pathogen which is common to all of us? The discussion is open.
Surveillance is very important. If we want to work on, I think surveillance is very important in this field of research.
Yeah, that is very important because when we conducted the tricycle, for example, it is very useful to have different data with the time, because the percentage of carriage of E. coli, for example,